Hello, wishing you a great day. Today we will be discussing about the physiology of urinary system part 2. At the end of this lecture, you should be able to discuss the filtration, selective reabsorption and tubular secretion within the nephron. We will be also discussing the role of kidneys in maintenance of extracellular fluid volume and acid-base balance. We will be then describing the process of micturation. Reabsorption. In this particular picture, you can see that most of the substances that is being filtered in the glomerular capillaries, which is filtered into the Bowman's capsule, 99% of substances will be reabsorbed back into the blood. Right here the filtration is taking place and here the reabsorption is taking place. The process of returning filtered material to the bloodstream is called as a reabsorption. This may involve transport proteins. Normally glucose is totally reabsorbed. That means 100% reabsorption is taking place for the glucose. Whatever glucose is being filtered here, glucose, the 100% glucose will be reabsorbed here. That means in the urine you don't see the presence of glucose. That is why under normal circumstances in a normal individual's urine you don't get to see the traces of glucose because it is 100% reabsorbed. So the presence of glucose is seen only in diabetes mellitus. Otherwise in the normal case you will not be seeing this. Secretion. It is the process by which some materials are added to the filtrate and it is added to the lumen of the kidney from the blood. So you can see that secretion is taking place. This is the bloodstream, this is the capillary, peritubular capillary. From peritubular capillary you can see that the C, this which is marked by C, this arrow, it is moving to the tubules. So substances are secreted back from the blood into the tubule, into the filtrate. Active transport, usually of toxins and foreign substances, will be done. One of the example is penicillin. Penicillin is always secreted from the blood into the tubules. Excretion. Loss of fluid from the body in the form of urine, and you can see that the excretion is the last part which is happening. Excretion, so whatever after reabsorption and after secretion, then the amount of fluid which is lost from the tubule, that is the excretion. So if you want to write it as a formula, we can write it like this: amount excreted is equal to solute filtered plus amount secreted minus amount reabsorbed. So you will get how much amount is excreted out of the transport of sodium, chloride and water. Sodium, water and chloride reabsorption occurs in proximal tubule, including proximal convoluted tubule and proximal straight tubule. When we discussed about the nephron, we saw that the proximal convoluted tubule is a coiled structure, the convolutions, which is a coiling, whereas proximal straight tubule doesn't have the coiling. So both of these things, proximal convoluted tubule PCT and proximal straight tubule PST, are together known as the proximal tubule. 65% of the sodium, chloride and bicarbonate and potassium will be absorbed in this particular phase, and 100% reabsorption of glucose will take place. It also secretes organic acids, bases, hydrogen ions into the tubular lumen. So some amount of secretion is also taking place in the proximal tubule. Sodium, chloride and water reabsorption in the proximal tubule. The sodium potassium ATPase, the major force for the absorption of the sodium chloride and water. So here you can see that the sodium potassium ATPase, which we have already learned before, it is the same sodium potassium ATPase, which means it is an energy dependent active process, which is acting as the major force for reabsorption of sodium chloride and water. In the first half of the proximal tubule, the sodium is reabsorbed by co-transport along with glucose, amino acids and other solutions. So the first half of the proximal tubule, what you will see is sodium will be reabsorbed along with glucose. So glucose as a co-transport. So both the glucose and the sodium will be going in the same direction. So a carrier molecule will be used there, and through that carrier molecule, both sodium and glucose will get attached and it will be moving. So it can be sodium along with the glucose, or it can be sodium along with the amino acids or any other solutes. In the second half of the proximal tubule, sodium is reabsorbed mainly with the chloride ions. Along with the chloride ions, it is reabsorbed. So this is a diagrammatic representation of the various process of reabsorption which is taking place in the nephron. So you can see, you can appreciate that here sodium ions are reabsorbed by active transport. Negatively charged ions are attracted to positively charged. Now what will happen is, from the tubule, this is positively charged ions, it is moving into the tubules, right? It is moving into the tubule. Sodium is moving into the tubule. It is a positively charged ion. So this will be attracted by the chloride ion because it is negatively charged. So chloride also follows the sodium, because when sodium concentration increases here, it will start dragging the chloride from here. So negatively charged ions are attracted to the positively charged ion. So when sodium enters here, it will be dragging the chloride. So here NaCl is formed, common salt, that is formed inside the blood. When this increases, it will be increasing the solute concentration within the plasma. As a result, the osmotic pressure increases, right? The number of osmotically active substances will increase in the plasma. When osmotic pressure increases, that will start dragging water into the blood, right? So you can see that as concentration of ions or solute increases in plasma, osmotic pressure increases. So osmotic pressure will be increased here. Then as a result, water will be pulled from the tubules into this particular space of where the plasma is there, where the blood is there, right? So water moves from the proximal tubule to the capillary by the process of osmosis, right? So this will be happening. So here when sodium moves, first sodium moves, then chloride will be following. So here we will have NaCl, then the osmolarity increases here. As a result, the water will be dragged. When water is dragged, what will happen? When water is dragged, here this particular area will be isoosmotic. That means equal number of sodium chloride and water is moving. So this particular fluid within this particular segment is said to be isotonic. See here, isoosmotic or isotonic tubular. Why it is called isotonic? Initially you can expect that when sodium and chloride is moving, the tonicity will be reduced here, right? Because water is still there, which will make it diluted. But as a result of high osmotic pressure here, the water also moves, equal amount of water also moves. So this particular fluid here is called as isotonic tubular fluid, because the tonicity remains unchanged, because equal amount of the salts and equal amount of water will be moving. That's the reason. Sodium and water transport in the loop of Henle. The loop of Henle consists of three functionally distinct segments: the thin descending segment, the thin ascending segment, and the thick ascending segment. So one of this is going down, which is the descending segment, a thin portion of the descending segment, a thin portion of the ascending segment which is going up, a thick ascending segment which is the one which is going up. The thin descending loop of Henle. Here the permeability is different between the ascending limb and the descending limb of the loop of Henle. Please note that the descending limb of loop of Henle, see the thin descending limb of loop of Henle, that means the one which is going down. The thin descending limb of loop of Henle is highly permeable to water and moderately permeable to most of the solutes, but has few mitochondria and little or no active reabsorption. That means it is highly permeable to water but not to the solutes. The thick ascending limb of loop of Henle reabsorbs about 25% of the filtered load of chloride, sodium and potassium, as well as large amount of calcium, bicarbonates and magnesium. This segment also secretes hydrogen ions into the tubules. So here you need to remember that there is a difference between the thin descending loop of Henle, which is highly permeable to water, but this one is not permeable to water. This is permeable to the solutes. Thick ascending limb is permeable to the solute, not to the water. So there is a striking difference. So this is permeable to the water, but here you see it is permeable. The thick ascending limb of loop of Henle is permeable to the solutes, reabsorb 25% of the filtered loads of sodium, chloride and potassium, as well as large amount of calcium, bicarbonate and magnesium. And there's also secretion which is taking place. The hydrogen ions will be secreted into the tubules. Renal threshold for glucose is the plasma level at which the glucose first appears in the urine. The renal threshold is about 200 milligram per deciliter. Please remember, deciliter means per 100 ml. So it is 200 milligram per 100 ml of arterial plasma, which corresponds to a venous level of about 180 milligrams per 100 ml. So that is the renal threshold. That means if the arterial plasma level of glucose is 200 milligram per deciliter, you can expect that it will start excreted out in the urine. You will start seeing the high levels of glucose in the urine. Hydrogen secretion and bicarbonate reabsorption. Hydrogen secretion is through secondary active transport. Again, what is a secondary active transport? A secondary active transport always utilizes the concentration difference that was created by a primary pump like sodium, potassium, ATPase pump. Mainly at the proximal tubules, loop of Henle and early distal tubule, you get to see this hydrogen secretion happening. More than 90% of bicarbonate is reabsorbed by passive method in this manner. Distal convoluted tubule and collecting duct. Tubular secretion is for getting rid of those substances like potassium, hydrogen, urea, ammonia, creatinine, and certain drugs. If these substances are accumulated at high levels in the plasma, that can create a toxic effect. Secretion of H ions also help in maintenance of blood pH. Urine formation, tubular reabsorption in the distal convoluted tubule. Kidney has remarkable ability to concentrate the urine or dilute the urine depending upon the conditions of the body. If a situation arises where the body has to conserve the water, then these hormones are released. Aldosterone, an antidiuretic hormone. So these hormones will make sure that it preserves your body water content and extracellular fluid volume will expand. ADH will make sure that it will reabsorb the water, and aldosterone will make sure that it will reabsorb the sodium. So when sodium is reabsorbed in larger amounts, it will definitely drag chloride. When NaCl is there in the plasma, it will also drag water. So that's an indirect way by which the extracellular fluid volume can be increased. So that depends upon the need of the body. So it is subjected to hormonal control. Around 36 liters of water or the plasma enters the DCT, from which the water content is being reabsorbed. Collecting duct at the papilla of the pyramid, ducts join to form 15 larger papillary duct which empty into minor calyces, and this is also having a very important role in the conservation of the body. Urine formation, tubular secretion, chemical extracted from peritubular capillaries. So you can see that from the capillaries, these substances like urea, drugs, ions, potassium, etc. enters into the distal tubule and collecting duct, and it will be moving out in the urine. Waste removal, especially the nitrogenous waste in larger amount, can cause toxic effect to the cells. It is having an important role in the acid-base balance. Secretion of ions and bicarbonates will be providing for the acid-base balance of the body. Regulation of pH is very much essential. Collecting duct receives several nephrons. It reabsorbs water. Concentrate urine becomes up to 4 times more concentration. So you need to remember that kidney is having remarkable ability to concentrate the urine or to dilute the urine depending upon the body situations or the circumstances. Concentration of the urine depends upon the body's state of hydration. So this picture will give you a summary of the tubular reabsorption and secretion. So all those things are what we have already explained. So this is just for an understanding. Remember that the last part, the collecting duct and the distal tubule, this is the area which is regulated by the hormones like aldosterone and ADH. Whereas in the proximal tubule or loop of Henle, the hormones doesn't have any role because there is no receptors for these hormones. That is the reason you will be seeing that the collecting duct and the DCT, they are usually called as a regulating segments because they are regulated by the hormones. Acid-base balance. All biochemical reactions are influenced by pH. Therefore, acid-base balance is closely regulated. The normal pH is within the range of 7.35 and 7.45. Whenever the pH of the arterial blood increases more than 7.45, it is termed as alkalosis, and when the pH of the blood is less than 7.35, it is called as acidosis. Some of the reasons that can lead to acidosis are release of phosphoric acid from phosphorus containing proteins, the production of lactic acid as a result of anaerobic respiration, formation of ketone bodies by fat metabolism, and also carbon dioxide transport in the blood in form of bicarbonate, which liberates ions. Abnormalities of acid-base balance. All classes are classified under respiratory and metabolic disturbances. Respiratory acidosis and alkalosis. It is caused by the failure of respiratory system to perform the pH balancing role. Here the most important part you need to remember is here the problem is lying at the respiratory system. If the lungs is incapable of expelling the carbon dioxide will accumulate in the blood and then it will increase the acidic content. When carbon dioxide is more, it forms the carbonic acid and the acidic content in the blood rises. And this leads to acidosis, and this can be called as a respiratory acidosis. If too much of carbon dioxide is released outside of the lung, that means if more amount of carbon dioxide is going out of the lungs as a result of hyperventilation, that can result in alkalosis. The single most important indicator is blood PCO2. Metabolic acidosis and alkalosis, on the other hand, it has nothing to do with the respiratory system. The other systems, if there is any problem, such as in the case of blood or in the case of kidney, if there is any problem that can alter the pH, alter the acid-base balance, those kind of situations are referred as metabolic acidosis and metabolic alkalosis, or abnormalities other than caused by the partial pressure of carbon dioxide levels in the blood, indicated by abnormal bicarbonate levels. Respiratory acidosis and alkalosis. Most important indicator of adequacy of respiratory function is the PCO2 level, or the partial pressure of carbon dioxide level, and the normal levels are between 35 and 45 millimeters of mercury. Some of the common causes of acid-base imbalances are respiratory acidosis, when the carbon dioxide level, the partial pressure of carbon dioxide rises above 45 millimeters of mercury due to a reduction in the ventilation or gas exchange. So here the problem lies at the level of the lungs, right? So the lung is not able to remove that much amount of carbon dioxide from the lungs. So in this case, what happens? Carbon dioxide will start accumulating in the blood. It forms carbonic acid, which is acidic. So it will lead to a respiratory acidosis, because the acidosis as a result of a failure of the respiratory system. Carbon dioxide accumulates in the blood, characterized by falling of blood pH and rising of PCO2. Respiratory alkalosis, when the PCO2 falls below 35 millimeters of mercury. One of the common cause for this is hyperventilation. Hyperventilation is too much of increase in the respiratory rate and the depth of breathing. That is called as a hyperventilation. In such cases, there is washout of carbon dioxide from the respiratory system, from the lungs. So this also happens during stress and also pain, where the person undergoes hyperventilation. There is also some cases where there is a problem with the medullary respiratory centers. If there is any problems, that can also give rise to such situations of hyperventilation. Carbon dioxide eliminated faster than it is produced. Metabolic acidosis, low pH and bicarbonates. Some of the causes for this is ingestion of too much of alcohol. When the person consumes too much of alcohol, so here what will happen? The acid content will be increased in the blood, but here remember that this has nothing to do with the lungs. Here adding of carbon or of acidic products is due to other systems, like in this case blood. If you are consuming alcohol, that is directly the alcohol content is increased, and then it will form the acetic acid, and the acid content will be increased in the blood. Excessive loss of bicarbonate, like in the cases of persistent diarrhea, when a patient has too much of diarrhea, he or she will lose so much of bicarbonate. So it will be when bicarbonate is lost from the body, the acidic contents will be dominated in the blood. So it will lead to a condition of acidosis. So here, because the lungs is not involved, we call this as a metabolic acidosis. Accumulation of lactic acid as a result of exercise. Too much of exercise can lead to accumulation of lactic acid, or in the case of circulatory shocks, cardiovascular shocks. Ketosis, like in cases of diabetic crisis or starvation, and also one of the most common is the kidney failure. When the patient has a kidney failure, that will be resulting in accumulation of lactic acid, then it will lead to metabolic acidosis. Metabolic alkalosis, much less common than metabolic acidosis, initiated by rising blood pH and bicarbonate. Causes like vomiting of acid contents of the stomach. As you know that in the stomach there is hydrochloric acid which is acidic. So too much of vomiting can remove these acidic contents from the stomach, and it can result in metabolic alkalosis. Or excessive intake of alkalis like antacids. Antacids are those substances, those medications that is taken against acidity. When the person is having acidity, we are advised to take antacids, but uncontrolled consumption of antacids can increase the amount of bases or alkali in the blood, and it can result in alkalosis. Effects of acidosis and alkalosis. Acidosis, when the blood pH is below 6.8, it can finally lead to depression of central nervous system, which can cause a coma, which is the unconscious state, and then leading to death. Alkalosis, blood pH above 7.8, there might be excitation of the nervous system. Muscles will undergo tetany, uncontrolled contractions without relaxation, extreme nervousness, convulsions, and death often occurs as a result of respiratory arrest. There might be laryngeal spasm and death of the individual. Compensations for abnormalities of acid-base balance. Respiratory system attempts to correct metabolic acid-base imbalances. Changes are brought about by changing the respiratory rate and depth of breathing. Kidneys will try to correct the respiratory acid-base imbalances. Respiratory compensation. Metabolic acidosis. High H plus ion as a result of hypercapnia. Hypercapnia means too much of carbon dioxide levels in the blood, which can give rise to excess amount of H ions. And this will stimulate the respiratory centers, and thereby it will increase the rate of respiration and depth of respiration. Blood pH below 7.35 and bicarbonate level is low. Here you can see that the rate and depth of breathing is elevated, then the carbon dioxide is eliminated or washed off from the respiratory system, then PCO2 falls below the normal range. Metabolic alkalosis. High pH over 7.45, elevated bicarbonate levels. Slow, shallow breathing, allowing carbon dioxide accumulation in the blood. PCO2 rises above 45 millimeters of mercury. Renal compensation for respiratory acid-base balance. For the acid-base balance, kidney and lungs together functions in a cooperated manner to maintain the acid-base balance. So both these organs have a major role in maintaining the acid-base balance. respiratory acidosis. Hypoventilation causes elevated PCO2 indicated by above normal value of plasma HCO3 minus, that is bicarbonates. Kidney will be compensating by excreting excess amount of H ions, because here the case is due to it is acid, right? Acidosis. So H plus ion has to be eliminated from the body. So what kidney will do in this kind of situation is that it will be excreting more amount of H ions and it will be retaining the bicarbonate ion. So kidney is selective in its function that it can eliminate the bicarbonate or it can eliminate the H ion depending upon the need of the body. Respiratory alkalosis. Here in these cases, if it is a problem with the lungs, what it will be doing is, here the situation is there is a low PCO2 and high H ions indicated by below normal value of plasma bicarbonates. So kidney will be trying to compensate this particular issue by retaining more of H ions and excreting more of bicarbonate, because here the problem is because of too much of alkali or bases in the blood. So we need to remove so much amount of bicarbonate. So you can see that kidney will be excreting so much amount of bicarbonates, at the same time it will be retaining the H plus. So H plus secretion reduces and bicarbonate secretion increases. So that is the kind of a compensatory role of the kidney, which is in towards the preservation of the acid-base balance. This picture will give you an idea about the reabsorption of bicarbonate, which is coupled to the secretion of H plus. So the first event, what is happening is carbon dioxide in combination with water in presence of the enzyme carbonic anhydrase, it forms carbonic acid. H2CO3 is carbonic acid. As soon as this is formed, it will be splitting into H plus and HCO3 minus. This will be split into two ways. Then the H ions will be secreted into the filtrate. It is going to the filtrate. So this is a tubular lumen. So it is going, and the H ion is going to the filtrate. Then what happens? For each H ions that are secreted here, you can see that it is compensated by the movement of bicarbonate into the blood. It goes into the blood. Remember, by two ways this bicarbonate can move. So bicarbonate will be taking, either it can go as a symporter or it can go as an antiport along with chloride. What is a symport? So symport, when two different ions are moving in the similar direction. If for example you see sodium is moving from here to here, and at the same time the same direction the bicarbonate is also moving, and they are sharing a common protein channel, right? A common protein channel is shared between the sodium and the bicarbonate. That kind of mechanism is called as a symport. So here you can see that symport mechanism. So along with sodium, it is following the symport. But when you are seeing when it is moving along with chloride, it is the antiport. See, chloride is moving from this direction, and this is the protein which is used. And you can see that bicarbonate is moving in the opposite direction. Bicarbonate is moving from here to here into the blood, whereas chloride is moving in this direction. So this is an example for an antiport mechanism. So it will adopt any of these two mechanisms. The secreted H ions will then combine. See, secreted H ions will then combine with bicarbonate to form the carbonic acid. Again, it will be broken down into water and carbon dioxide will be then diffusing into the tubule, and this will be triggering the this particular reaction, and it will be forming like a cycle. So this is what happening in the renal cells in the regulation of H ion secretions. These are the steps involved in the urine formation. The first and foremost, it is happening in the glomerulus, which is the glomerular filtration, which creates a plasma-like filtrate into the blood. Then there is tubular reabsorption, where necessary substances are reabsorbed by the kidney tubules. It removes the solutes from the filtrate and returns that back to the blood. Tubular secretion, it is this process by which additional substances are added into the filtrate, removes additional waste from the blood, adds them to the filtrate. Then comes to the last part, which is the water conservation, which removes water from the urine and it will be returning back to the blood. So it is the process by which the water conservation is taking place. So the urine will be concentrated here. Urine volume. Normally 1 to 2 liters per day of urine is formed. More than 2 liters per day is termed as polyuria. Some of the causes for polyuria are excessive fluid intake, conditions like diabetes, or some of the drugs which are called or which can induce diuresis. Less than 500 ml per day is termed as oliguria, which are causes. Some of the examples are kidney diseases, dehydration, circulatory shock, prostate enlargement are some of the causes for oliguria or less urine output, less than 500 ml per day. Less than 100 ml per day is anuria. This is seen in emergency crisis like excessive cardiovascular shocks, like hemorrhagic shocks. When the person has lost so much amount of blood, the patient will have a complete shutdown of urine formation, which is called as anuria. Urine storage. Ureters carry urine from kidney to the urinary bladder by the process of peristalsis, or rhythmic contraction of the smooth muscles. Pressure from the full bladder compresses the ureter and prevents back the flow. If it is a full bladder, the bladder seems to be spherical in shape and extends into the abdominal cavity, and it can hold about half liter of urine, 500 ml. If the bladder is empty, it lies within the pelvis with shape like upside down pyramid. Urine excretes via the structure called urethra. Trigon is a triangular area seen inside the urinary bladder between the ureters and the urethra. They are prone for infections. Urine elimination. Urethra drains urine from the bladder. So you can see that this structure is the urethra, which will be draining the urine from the bladder to outside. There is a structure which is called internal urethral sphincter. This is the internal urethral sphincter, which is made up of smooth muscles, and they are having involuntary control. That means we don't have any control over it because it is involuntary, because it is made up of smooth muscles. External urethral sphincter is this one, which is a skeletal muscle which is voluntary. That means the external urethral sphincter can be relaxed and constricted according to the wish and will of the individual. Provides voluntary control over voiding. So this is important. So there is a striking difference between internal urethral sphincter and external urethral sphincter. Internal urethral sphincter is smooth muscle and it is involuntary. External urethral sphincter is skeletal muscle and it is voluntary. Micturation. It is the process of emptying the urinary bladder. It is also known as voiding or urination. Two processes are involved. The bladder fills progressively until the tension in its wall rises above the threshold level, and then a nervous reflex called micturation reflex occurs. And that is the reflex which is responsible for emptying the bladder. The micturation reflex which is a very important slide to understand how the process of micturition or urination happens. Micturition reflex is an automatic spinal cord reflex. However, it can be inhibited or facilitated by the centers in the brainstem and cerebral cortex. The urge to urinate when the bladder contains 200 ml of urine. When the bladder is filled with 200 ml of urine, the urge for urination occurs. We will look into the finer details of the micturition reflex in the next slide. Micturition reflex. Here it is sensed by a receptor. The stretching of the bladder wall by urine is sensed by a receptor. It is sensed by a receptor, and that are stretch receptors. Efferent pathway, they are the pelvic nerves. Pelvic nerves carry the sensations of stretching. The center here, they are sacral spinal cord, which includes S2, S3 and S4. The sacral spinal segments S2, S3 and S4, they are the center involved in this particular reflex. So the efferent pathway is pelvic nerves, which will be carrying the stretching or distension of the bladder. The efferent pathway is pelvic parasympathetic and somatic pudendal. So somatic, when you hear the term somatic, means it is voluntary. Parasympathetic, which means it is involuntary. And effector structures will be the detrusor muscle. So the detrusor muscle, which leads to contraction, which will be through the pelvic parasympathetic nerves, whereas somatic pudendal will be functioning, or which will be innervating the external sphincter. And remember, the external sphincter is made up of skeletal muscles, so it will be under voluntary control. That is why this nerve is a somatic nerve, and the name of the nerve is pudendal nerve. Response: contraction of the detrusor muscle and relaxation of the internal and external sphincter. So this one will facilitate the urination. And higher centers involved in the urination process or a micturition reflex is pons and cerebral cortex, because in an adult individual, according to our wish and will, urination is possible. If the situations are favorable, the individual can micturate. So you can have that capacity to inhibit the micturition process by constricting the external urethral sphincter, because it is under voluntary control. So this is a picture which shows the micturition reflex. So you have to just see which are the centers involved, what are the efferent nerves, what are the efferent nerves, etc. And these are the references. Again, we will be having a live session to discuss the renal physiology part 1 and 2, so that your doubts and clarifications can be made in those sessions. And once again, thank you for listening.